Muy buenos días a todos y bienvenidos a la 14ª edición del Foro CIBERNED que celebramos en esta ocasión de forma virtual. Desde la última celebración del foro hemos sufrido la irrupción de una pandemia que ha afectado a nuestra sociedad de forma muy profunda, generando una ola de sufrimiento, de muerte, de empobrecimiento sin precedentes. A nivel sistémico, el virus está dejando al descubierto la fragilidad de muchas de nuestras estructuras fuertemente consolidadas, no solamente en el ámbito asistencial, sanitario, sino también en el ámbito académico y científico. Y además, ya inmersos en esta segunda ola epidémica, estamos observando, los datos indican que los factores de vulnerabilidad individual a nivel social, sanitario y económico están teniendo un peso cada vez mayor. Y ante esta situación, todas las voces indican que el camino de retorno está en la ciencia, todas las miradas están posadas en la ciencia, esperando una vacuna o un tratamiento eficaz, y se invoca a la ciencia en todos los foros, en foros no científicos, en los que frecuentemente no se tiene en cuenta elementos fundamentales de apoyo, de estabilidad, de continuidad. Y es precisamente en foros como este, en foros científicos compuestos por personas que han dedicado muchos años de su vida a la ciencia y que entienden las necesidades y también las oportunidades que nos aporta la ciencia, donde es más importante que nunca compartir las necesidades de la sociedad y también los resultados a los que se están llegando. Un buen ejemplo de esto serán los resultados que se vayan a presentar a lo largo de los próximos 2 días. Porque las enfermedades neurodegenerativas continúan siendo uno de los mayores retos a los que nos enfrentamos como sociedad. La pandemia no ha hecho disminuir la relevancia de la lucha contra las enfermedades neurodegenerativas, más bien al contrario. La infección por SARS-CoV-2 ha endurecido las consecuencias devastadoras de las enfermedades neurodegenerativas, tanto de forma indirecta, debido a, como saben, a la mayor dureza de la enfermedad en pacientes de edad avanzada, como también de forma directa en elementos que están en este momento en evaluación y en estudio. Por una parte, el potencial factor de la infección por SARS-CoV-2 en el inicio y en la progresión de enfermedades neurodegenerativas y neuropsiquiátricas vinculadas a factores inflamatorios. Y, como saben también, el estudio del posible efecto que puede tener las medidas de distanciamiento social sobre la evolución de las enfermedades neurodegenerativas. Nuevas preguntas que requieren respuestas y toda la experiencia de nuestra comunidad científica y sanitaria. Y junto a estos nuevos retos es imprescindible redoblar los esfuerzos para activar de forma decidida una respuesta coordinada para mejorar el diagnóstico, retrasar la progresión, aliviar el sufrimiento y también para intentar buscar un tratamiento para muchas de las enfermedades neurodegenerativas que aún no tienen una cura. Y para ello, quiero decir que no es suficiente como un sumar fuerzas, es necesario multiplicar. Con esto quiero decir que CIBERNED está llamado a ser más que la suma de sus grupos, más que la suma de sus investigadores. Necesitamos ser un factor multiplicador. Precisamente este es el espíritu con el que se creó CIBERNED. Esta es la misión hacia donde caminamos: favorecer grandes proyectos con capacidad de transformación apoyados en sinergias. Y permítanme pasar a cada uno a lanzar un mensaje a cada uno de los investigadores, cada una de las personas que forman parte de CIBERNED. Este foro es una oportunidad extraordinaria para celebrar la perseverancia y el compromiso de cada uno de vosotros, cada uno de los que habéis dedicado vuestra vida entera a luchar contra las enfermedades neurodegenerativas. Sabemos, y ahora la sociedad cada vez es más consciente, que la ciencia es el único camino y que además es posible avanzar y encontrar soluciones para situaciones, para enfermedades que hace poco tiempo creíamos incurables, intratables. No cabe duda que este camino es de enorme dureza y por eso quiero expresaros mi más sincero agradecimiento y animaros a que, a pesar de todas las dificultades que se han incrementado este año, continuéis esta misión tan necesaria. Mis últimas palabras son de agradecimiento personal. Quiero focalizar este mensaje de agradecimiento en una persona que ha sido el alma de CIBERNED a lo largo de los últimos 9 años. En octubre de 2011, el profesor Ávila aceptó con la generosidad que le caracteriza el puesto de director científico en CIBERNED. A lo largo de todo este tiempo ha dirigido el Centro de forma brillante, contribuyendo de forma decisiva a alcanzar la madurez científica de CIBERNED. Gracias al profesor Ávila, en este momento se dan las circunstancias idóneas y el nivel de madurez suficiente en la organización para avanzar también en la renovación de sus órganos de gobernanza, cumpliendo el mandato recogido en los estatutos de CIBERNED. Como saben, los estatutos de CIBERNED y de todas las áreas temáticas de Ciber incluyen la renovación del director científico cada 4 años. Y en este 14º foro, en esta reunión de científicos de CIBERNED del más alto nivel, es una ocasión idónea para agradecer y reconocer todo el esfuerzo, la pasión y la dedicación que ha depositado el profesor Ávila en CIBERNED y que sin ninguna duda perdurará en los años venideros. Sin más, quiero desearles unas jornadas muy fructíferas. Realmente, viendo el programa, se puede percibir el nivel de la investigación que se hace en CIBERNED. También quiero transmitirles que, sin ninguna duda, la lucha contra las enfermedades neurodegenerativas continuará siendo una de las prioridades del Instituto de Salud Carlos III. Muchas gracias a todos.
Muchas gracias, directora. Buenos días a todos y bienvenidos al 14º Foro de CIBERNED. Para aquellos que no me conozcan, mi nombre es Miguel Medina, soy director científico adjunto de CIBERNED. Y antes de pasar a iniciar la primera sesión científica, querría hacer un par de consideraciones de tipo práctico para que todos tengamos claro en este foro tan peculiar y especial debido a las circunstancias que vivimos cómo va a funcionar digamos, desde el punto de vista logístico o práctico. Vamos a tener sesiones científicas con aproximadamente 3 ponentes. En la mayoría de ellas, los ponentes estarán conectados a la vez, en paralelo, pero solo uno de ellos entrará a intervenir en cada momento. Sí que me gustaría solicitarle a los ponentes un par de cosas, simplemente recordarles. Primero, que se ajusten al tiempo previsto. Segundo, que el que no esté participando, por favor, se asegure de que el micrófono esté apagado para evitar interferencias. Y tercero, los asistentes van a poder realizar preguntas a través del chat que la plataforma les ofrece. Estas preguntas llegarán por escrito durante todo el tiempo. Yo tendré aquí un acceso a esas preguntas y al final de cada sesión tendremos una sesión de discusión en la cual yo iré trasladando las preguntas a los distintos ponentes en función de cómo vayan llegando y el tiempo que tengamos disponible. También me gustaría recordarles que tenemos una plataforma con pósters. Creo recordar que hay aproximadamente 70 y tantos pósters disponibles para que todo el mundo que está inscrito tiene acceso a través de esa plataforma de póster, en la cual también hay la posibilidad de entrar en discusión con los autores de los pósters, ya sea a través de un chat privado o un chat público, en función de lo que decida cada uno de los participantes. Y animo a todos a que en los breaks y en las pausas, tanto de la comida como entre sesiones, pues entren en este chat y participen activamente en la discusión, especialmente la gente joven, de manera que intentemos hacer que este foro tan peculiar sea lo más parecido a un foro presencial de los que solemos organizar anualmente, y que la interacción sea tan amplia como suele serlo. Voy a simplemente ahora decir unas palabras repitiendo esto en inglés, por si hay alguno de los asistentes que no domine el español, y también les diré que las sesiones en principio serán en inglés, aunque hemos dado la libertad a los distintos ponentes de elegir el idioma en el que participen. Good morning, everyone. Welcome to the CIBERNED Forum. I just wanted to make a few comments before starting the first scientific session. The first one is directed to the speakers. I would just like to kindly ask all the speakers to be strict in the timing of the presentation, and also for those of them who are not participating at the moment, just to keep in the mute position the microphone in their computers. There will be 3 speakers per session, and then after the third talk, there will be a time for discussion. Everyone has the possibility to make questions through the platform. I will receive the questions here in writing, and I will transfer the questions to the speakers right at the end of the session. There is also the possibility to get into the poster platform. I'm sure you all have received the links to connect to these posters, and the posters platform has also a possibility of making questions and getting into discussion with the authors of the poster, whether that's in a public chat or through private ways. And I would like to encourage everyone, especially the youngers in CIBERNED, to get into this active discussion as much as possible. And well, after all this, I just want to get going with the first scientific session. This first session will be incorporating 3 speakers, Dr. Isidro Ferrer, Dr. Eva Carro, and Dr. Jesús Ávila. As I said earlier, the 3 will be in parallel in the session, but only one will be speaking at a time. I'll ask the other 2 to keep the microphone in mute to avoid any interference. And just insist again, there will be a period for questions at the end of the third talk. So without much delay, I will just want to introduce you the first speaker today and from the conference, which will be Dr. Isidro Ferrer from the Bellvitge Institute of Research. And he will talk about global glial tauopathy, neuropathological and molecular characterization. Isidro, you have the floor, please. Thank you.
Thank you. Hi, good morning. First, this is new for me, and this is in some way some kind of new novelty to make this kind of presentation. So perhaps I commit some errors or mistakes, and I would like to apologize at first for any inconvenience. Well, my talk is about global glial tauopathy, and this is a globular gliotauopathy, and this is a new disease. And this disease begins many years ago by the description of very individual cases, which were covered altogether at the beginning of the century. Further manifested in the form of consensus recommendations, and at this time, this globular gliopathy was considered a new entity. We have the possibility to study many cases. There is an echo. Well, we have the opportunity to study several cases before the consensus epoch, and then some others from this time onwards. So what I'm going to do is to analyze our cases in the context of what we know now at present, what we know as familial or globular gliotauopathy. So the other cases we have studied were studied by Dr. Erro, and they belong to two pedigrees with a number of patients with different pathologies. And these different pathologies or different clinical manifestations. These were characterized by corticobasal degeneration, progressive supranuclear palsy, frontotemporal dementia behavioral variant, and in addition, some cases had involvement of the motor system. So the main important point from the neuropathological point of view was the presence of typical globular incursions in astrocytes and oligodendrocytes. And here is the name of globular gliotauopathy, because the characteristic lesions in the astrocytes and in oligodendroglial cells. These are the pedigrees, pedigree 1 and pedigree 2, and this is the names of the patients. All of them in black were studied neuropathologica. So, and this represents the first large series of cases with mutations in the MAP gene. So one of patients showed involvement of cortical atrophy, another one atrophy of the hippocampus, the other or others global hypometabolism in the brain. So the clinical features differ from one patient to another, and the radiological findings also differ from one patient to another. This is a summary of the lesions, the neuropathological lesions. I don't want to go into detail, but just to name or to stress the typical lesions, the form of globular astrocytic inclusions, the globular oligodendroglial inclusions, which are typical and common in all the cases and are the hallmark of this disorder. In addition, there are neuronal inclusions, which are tau positive, and the involvement differs from one patient to another. So there is not a unique phenotype, but several different phenotypes. Well, these are the characteristics. This is one brain with atrophy. The other images show these typical globular inclusions here in oligodendroglial cells and astrocytes. Here is another in oligodendrocytes. And one of the most characteristic features is that these inclusions contain a perphosphorylated tau, which is 4R, but not 3R tau. Well, here is the picture showing the mutation, which in this case is P301T. And here is the characteristic Western blotting of these cases. So when comparing with patients with Alzheimer's disease, we show three typical bands at 68, 64, and 60 kilodaltons. These patients show two bands, which are typical of our tauopathy. These bands are 68 and 64 kilodaltons, and several small bands of lower small molecular weight, and some bands with truncated tau. Well, here is a summary of the alterations or the cases which are familial. And here are the different mutations presented in these cases. Here is our case presented by Professor Sarranth in 2005. And here are the new cases post-consensus area. So although the majority of cases or GGT cases are sporadic, there are also many familial cases which are due to different mutations in the MAP gene. Next, and perhaps is the most important point of this presentation, is the molecular characterization of GGT. So one finding or one thing is the identification or the recognition of a new entity, and the other one is to try to recognize the differences and the characteristics, the molecular and pathological characteristics in these cases. So we studied in the second part the molecular aspects of sporadic cases, one sporadic case and another inherited case due to mutation in K317M. And in addition, we had the other cases. And here, what we have here is there's an echo. Well, there's a feature showing the characteristics again of this entity with tau in neurons in glial cells, showing the typical characteristics of globular associative inclusions, and here again the other one with the globular inclusions oligonuclear cells, and the fact that all these inclusions are 4R and not 3R. In this other, and in the other picture, we can see conformation and alterations of this tau incorporated in deposits, oligomers in these deposits, and the presence of truncated tau at the carboxy terminal. So this is labeled by this antibody. Well, again, in another case, what you can see is these similar alterations, but with a particular morphology. For example, in addition to globular astrocytes, what we can see here is different astrocytes with different inclusions and different globular inclusions. So, in addition to the diversity in clinical and radiological manifestations, there is also diversity, heterogeneity in the neuropathological features. Another point, what is important, is the alterations in different glial cells. For example, what we have studied here is the expression of different astroglial markers. For example, IKL40. The other one, which is increased, aquaporin 4, which is increased, but a decrease in the expression of glutamate transporter 1 and of glucose transporter. So these astrocytes are not normal. They are normal, and these abnormalities manifested by different or an increase in this case or a decrease in particular markers. In similar way happens with mitochondrial pyruvate carrier 1, MPC1, which is decreased in patients with GGT. There is also an alteration in the precursors of oligodendroglial cells, and this is accompanied by a decrease in the expression of myelin markers, together with an alteration in axons in the wet matter. Well, when we are comparing or analyzing the results in more detail, we can see, for example, that there is a decrease in the ECP5 in globular astrocytic inclusions. And another important point is that these processes or these alterations are active in the sense that several kinases are localized with how the in this cell. What we have now next is to study the transcriptome of different genes related to astrocytes and oligodendrocytes. And we can see here that several genes, such as this one, GFP, Cal40, the connexin linked to astrocytes, are significantly increased, whereas the other one, mitochondrial carrier 1, is significantly decreased. This happens in the frontal cortex, and this is accompanied by an increased expression of NG2. But a trend to decrease in the expression of IL genes. When we compare the results in the frontal cortex with those in the white matter, what we can see is that there is, in contrast to the frontal cortex, a marked reduction in the expression of different markers of oligodendrocyte cells and of genes involved in myelin. So the alteration in astrocytes and oligodendrocytes have an impact on the function of the cells. We next analyze the transcriptome, the proteomic disturbances in the frontal cortex and white matter in these cases. We can see here is that there is a tremendous abnormality in the protein and phosphoprotein expression in the frontal cortex and in the white matter. And alterations in astrocytes, oligodendrocytes, and neurons, both in the frontal cortex and in the white matter. In addition to mRNA alterations, what we have is several protein alterations. These protein alterations are affecting proteins and proteins as well. So when we are using the study of biological function following the gene ontology, what we can see here is that several pathways are altered in these cases, in the frontal cortex and in the cortical white matter. So there is a tremendous alteration in the different pathways. And for example, what we here see is the protein network in frontal cortex, considering proteome. So this is a very important point, because what we consider typical of the, which is the phosphorylation, normal phosphorylation of tau. Phosphorylation is not specific of tau, but also involves several other proteins which are normally phosphorylated. And the point is the alteration in the frontal cortex of synaptic proteins. So, for example, this point has been very well studied in Alzheimer's disease, but synaptic pathology is not classic of Alzheimer's disease of natural death bodies. But here, for example, what we can observe is a tremendous alteration of proteins linked to synapses in the frontal cortex cases. In another case in which we have repetitive pathological alterations in the other mutation, K317T, showing similar alterations, and in addition, several mutations in the motor neurons of the spinal. And again, what we can see when we use Western blots is that there is an heterogeneity in the expression of different tau species in the different molecular layers of. So there is no pattern, but a pattern which differs among patients to another. What is typical for all of them is the presence of this band 64 kilodaltons, who represent the characteristic of 4-hour neuropathies. So this is a summary of the main alterations, neuropathological lesions, molecular neuropathological lesions, and here I will want to stress that we can see different or several proteins which are hyperphosphorylated in these diseases or in this group of diseases when compared to normal patients. So there is not only that, but many other proteins which are phosphorylated. The next point is to know whether this kind of tau in GGT has the capacity of seeing and spreading when injected or inoculated in mice. So what we have seen here, what we have done here is to inject sarcosine solutions from GGT inoculated into the campus corpus callosum and white type mice. So what we can see here is after 3 months or 6 months, how inclusions in neurons, the hippocampus, but also glial cells in the villia, in the corpus callosum, in the ipsilateral middle region and lateral region. And these inclusions are positive for our tau, but in contrast with what happens in humans, there is also 3 tau kind of inclusion. So, what is important is that we have produced some kind of tau, but in contrast to the human counterpart, we cannot see globular glial inclusions. So what we can see is inclusions in a few astrocytes running, but in oligoastrocytes in the form of polybar. And here is a similar representation in the cognitive payment, where we can see several cells, most of them mirror with all glial cells which contain tau. Here is another image of microscopy, confocal microscopy, showing the position in neurons, oligodendroglial cells, but not in astrocytes. Here, the important point is that this process is active because there is co-localization of tau deposits with an active form of P50 kinase. So this represents this kinase, or this suggests that this kinase is active and is linked to the production of phosphorylated tau. Here, there's another model using sporadic GT inocula and inocula from GT harboring other mutation, and there is a similar pattern. So there is an involvement of neurons here, again neurons, and here the lateral corpus callosum and contralateral corpus callosum. And the same happens with the other case. So we have also shown that it can be reproduced in vitro by using data to show the sense of self. There is a positive conversion, so these granules represent signal is using inocula from transgenic mouse model of the property. Here is the, and here is another property which is called alpha. And here we can see in vitro that this tau coming from GGT cases has the capacity of seeing in vitro and in vivo. In summary, what we can see here is that tau in GGT has the capacity of getting and spreading in a particular pattern, not reproducing exactly the pattern of human disease, but reproducing some kind of tauopathy. This is important because our interpretation is that it's important the tau of the planner, also the characteristic of tau of the house. And for this reason, we can see several points or several differences, such as, for example, the lack of difficult inclusion and the presence of PR tau in addition to 4 hours. As regarding, I would like to stress the first case we have studied, which was in a publication regarding other cases of primary progressive aphasia. So I would like to remark my thanks to all these people. The novel mutation, which was characterized by Professor Ferrer 10 years ago, And then the consensus recommendation. So all these people, we were working together to find a consensus recommendation to doctor the different work researchers in our lab, which defined different characteristics of down different zoopathies. And here, Dr. Carro and all this team who described it for the first time, new zoopathy, cause of globular leuropathy, and here, which is the molecular pathology of familial globular neuropathy, was published here in Acta Neuropathology. And I would like to thank all these people who permit the production of this study. And thank you very much for your attention.
With the audio, especially at the beginning of the presentation, I hope you can be could have been able at least to follow up the presentation by Isidro. And so now we're going to go for the second speaker in this session, which will be Dr. Carro from the Doce Octubre University Hospital. And she will talk about salivary lactoferrin as a biomarker for Alzheimer's disease, brain immunity interactions. So Eva, please, you have the floor.
Well, I'm going to talk about salivary lactoferrin as biomarker for Alzheimer's disease, brain immunity interaction. And well, clinically, Alzheimer's disease is characterized by early memory deficits, followed by a decline in other cognitive functions. And pathologically, Alzheimer's disease is characterized by amyloid beta accumulation as amyloid plaques, hyperphosphorylated tau as intracellular neurofibrillary tangles, and chronic inflammation followed by the loss of neuronal cells, mainly in cerebral cortex and hippocampus. Despite the advantage in the knowledge of the Alzheimer's disease pathology obtained in these last decades, the mechanisms of action involving the pathology of Alzheimer's disease are still not completely understood. Although the toxicity of amyloid beta is fully accepted, there is a continued debate about the role of amyloid beta, if it is a cause or if it is a consequence, and the amyloid cascade hypothesis remains under controversy and debate. In the last few years, many studies have been published suggesting that brain infections with pathogens as bacteria or viruses may play an initiating role in amyloid plaque formation and the development of Alzheimer's disease. Neurodegenerative diseases, including Alzheimer's or Parkinson's diseases, are speculated to result from a combination of genetic and environmental factors, a concept commonly known as a multiple effect hypothesis. This hypothesis also proposes and reflects the complex and multifactorial etiology of these neurodegenerative diseases. It's not a new hypothesis. Oscar Fischer personally proposed that infections may play an important role in Alzheimer's disease already more than 100 years ago. More recently, the studies of Dr. Rodizaki revealed a potential link between infections and Alzheimer's disease pathology, and in particularly, she showed the presence of herpes virus in the mainly in the cerebral cortex of patients suffering Alzheimer's disease. In 2010, a hypothesis was postulated suggesting that brain infections with bacteria or viruses may play a crucial role in the amyloid plaque formation and the development of Alzheimer's disease. These infections, bacteria or viruses infections, lead to a persistent inflammatory stimuli regulated by cytokines that are known to induce stress and alter the immune response. This inflammatory response to a previous systemic infection may indirectly lead to an upregulation of the amyloid beta production and accumulation, and thereby initiating the amyloid effect. Indeed, as an example, I showed you this very recent study published in Nature a couple of weeks ago, where authors describe a protein, an interferon, produced by reactive astrocytes in response to a previous innate immune response. And this protein is able to modulate gamma secretase and then the production of amyloid beta peptides. Antimicrobial peptides are an essential component of the innate immune system against infections. These antimicrobial peptides and proteins are produced by many types of cells, phagocytes, epithelial, and endothelial cells, and represent an important defensive element inducing a broad spectrum of antimicrobial effects against many pathogens such as bacteria, fungi, protozoa, yeast, viruses. And these proteins and these antimicrobial peptides and proteins may directly act on the killing these pathogens by the interaction at the membrane, and they also are able to act via indirectly actions, modulating the response of immune cells. The role of antimicrobial peptides has been in these last years, and in fact, these peptides, these antimicrobial peptides, are produced and secreted from a number of secretory glands, including salivary glands. There are numerous defensive proteins present in saliva, but all of them, lactoferrin is the most abundant salivary antimicrobial peptide protein in this case, and represents a first defensive line with a wide spectrum of important biological functions, including antimicrobial section. Lactoferrin is an iron-binding protein belonging to the transferrin family, and it expresses in all the fluids, particularly in exocrine fluids, as tears, and as I mentioned in saliva. Lactoferrin has a wide variety of physiological functions, including antimicrobial functions against pathogens as bacteria, fungi, etc. But also this protein exerts an important anti-inflammatory action, modulating the production of pro-inflammatory cytokines. Lactoferrin also has antioxidant functions, modulating intracellular oxidative stress and even mitochondrial function, and other functions as immunomodulatory, anticarcinogenic, and enzymatic activities. In a recent study, we have proposed salivary lactoferrin as a novel Alzheimer's disease diagnostic biomarker. Here in this study, we found reduced salivary lactoferrin levels in patients with mild cognitive impairment and Alzheimer's disease compared with control healthy subjects. Due to the relevance of these findings, we decided to continue and to perform a new study, with the aim to evaluate the diagnostic utility of salivary lactoferrin in a well-characterized cohort of Alzheimer's disease and frontotemporal dementia patients, in order to assess the salivary lactoferrin specificity and its relationship with the pathology of Alzheimer's disease. Indeed, in this study, we used two independent cohorts, and the demographic and clinical data of the participants in both cohorts are shown in these tables. In this study, the protocol used include the salivary sample collection, levels of salivary lactoferrin were determined using a commercial and specific human ELISA kit, and amyloid PET scan and ApoE genotyping were carried out in all participants. And here I would like to show you the main results we obtained in this study. In the cohort 1, we found that salivary lactoferrin levels were significantly decreased in Alzheimer's disease and mild cognitive impairment groups, both of them PET positive, compared with the control group. We didn't find any differences between these groups, mild cognitive impairment and Alzheimer's disease. And regarding the frontotemporal dementia PET negative group, we found even a low increase in the levels, in the salivary levels of lactoferrin compared with the control healthy subjects. In the cohort 2, we also found a significant reduction in the salivary lactoferrin levels in the mild cognitive impairment PET positive group compared with the control healthy subjects, and also compared with the mild cognitive impairment PET negative group. Our results, these lactoferrin results, were also analyzed with using regression models, and here we can see that using the Jordan index, that the optimal cutoff point to differentiate mild cognitive impairment Alzheimer's disease PET positive patients with the control healthy subjects was 5.63, and the area under the curve was 0.95, with a sensitivity around 87% and a specificity more than 91% in the cohort 1. And in the cohort 2, in the validation cohort 2, using the same cutoff, we found, we obtained area under the, well, again, to differentiate the mild cognitive impairment PET positive compared with the healthy control subjects. The area under the curve was 0.93, sensitivity 87%, and the specificity more than 92%. Then, as conclusions from this study, salivary lactoferrin levels are significantly reduced in Alzheimer's disease patients, but not in patients with other dementia, such as frontotemporal dementia, suggesting a specific amyloid beta related link. I would like to tell you that even in our previous study published in 2017, we also found that lactoferrin levels in saliva from Parkinson's disease patients were not reduced. The presence of a positive amyloid PET signal is highly associated with reduced levels of salivary lactoferrin. Mild cognitive impairment without evidence of Alzheimer's disease pathology. I mean the mild cognitive impairment group with PET negative continues to be a mixed and frequently poor defined group. And here in this study, lactoferrin showed high variability levels in this group, in these mild cognitive impairment PET negative patients, reflecting their biological diversity. And we propose that salivary lactoferrin represents one of the main and first lines of defense against pathogens, and that low lactoferrin levels in saliva may denote its hypothesis, our hypothesis, that low lactoferrin levels in saliva may denote a reduced protection, exacerbating the risk of Alzheimer's disease. According to the Food and Drug Administration, an ideal biomarker is thought to be specific, sensitive, predictive, robust, simple, accurate, inexpensive, and should ideally be measured in standard biological source. Then we consider that salivary lactoferrin meets all these conditions. But why why salivary lactoferrin levels are reduced in these patients, in patients with marked cognitive impairment and sporadic Alzheimer's disease. The most accepted or credited pathological hypothesis of Alzheimer's disease in these last 3 decades is the amyloid cascade hypothesis. But this theory has increasingly been debated, mainly due to the negative results from the anti-amyloid clinical trials. And recently, other hypotheses have been proposed, although none of them have been widely accepted. In this study, in this recent article, we discuss the hypothesis that low salivary lactoferrin may reflect dysfunction in the immune system in sporadic Alzheimer's disease. Here we debate, in this study, we debate the relation and interaction between the nervous system and immune system, as both of them work in coordination in host defense. And as many authors suggest, the relationship between these two systems are bidirectional and very complex, but far from completely understood. In Alzheimer's disease, amyloid and tau accumulations may be found and observed in different brain areas, including hypothalamus, affecting its normal functioning. Since hypothalamus regulates basic body functions, hypothalamic dysfunction induced by these protein accumulations may result in abnormality and alterations in many functions, including immune system dysfunction. Thus, we propose that salivary lactoferrin may decrease due to a downregulation of the immune system associated with the hypothalamic alterations in Alzheimer's disease. Moreover, salivary protein secretion is controlled by parasympathetic nerves connected to the hypothalamus. The secretion of lactoferrin is regulated by cholinergic parasympathetic nerves that release acetylcholine that joins muscarinic receptors and evoking the secretion of saliva by acinar cells from the salivary gland. Thus, the hypothalamic dysfunction in Alzheimer's disease can directly involve salivary gland dysfunctions, including a reduction in the production and secretion of lactoferrin. However, although it is clear the complex relation between the nervous system and the immune system, it is far to be completely understood. And understanding this complexity is clearly critical for designing intervention strategies in the prevention of Alzheimer's disease. Then, in summary, Alzheimer's disease is a complex multifactorial syndrome, and mechanisms are still uncompletely elucidated. As infections may upregulate amyloid beta, as I showed you previously, antimicrobial peptides may contribute to amyloidosis progression and neurodegeneration in Alzheimer's disease. salivary lactoferrin seems to have a very good diagnostic performance to detect Alzheimer's disease, and our findings support the potential or the possible utility of salivary lactoferrin as a new non-invasive and cost-effective Alzheimer's disease biomarker. And the deficiency of salivary lactoferrin in Alzheimer's disease might facilitate oral pathogen infection and proliferation and the expansion of these pathogens to the nervous system. Well, I'm afraid that I only recommend healthy lifestyles, and I would like to thank all members of my group and the institutions involved in this study. Thank you so much.
Thank you very much, Eva, and thanks for sticking to the allocated time. Before we get to the next speaker, let me just remind you that you can all have post questions through the chat that you have in your platform to any of the three speakers, and I will get the chance to see those questions here and make those questions directly to the speaker at the end of the session. So please don't refrain yourself of asking questions. Hopefully the audio problems that we have in the previous talk has been improved in this last one. At least I'm not getting that many messages regarding that, but hopefully we will be in good shape for the next of the sessions and so on. So the next speaker in this session will be Dr. Jesús Ávila from the Center for Molecular Biology in Madrid, and he will talk about in vivo reprogramming increases adult neurogenesis and improves memory in mice. So Jesús, you have the floor, please. Go ahead.
Okay, first I would like to thank our director of the Institute, Carlos III, for her kind words to me. Second, I will start with my talk, and the talk is related about the development of human life, mainly focusing to aging. Mouse is a good system, it's a good model to study development and aging. And it is a dogma indicating that the development in mammals starts with a single cell that after several divisions and proliferation of these cells and followed by some differentiation ends in the formation of the whole organism. This mechanism is, as suggested by the dogma, unidirectional and is not way back to start to the cells, to the first cells. However, Yamanaka indicated at the beginning of this century that it's possible to, from a differentiated cell, to reprogram an embryonic-like pluripotent cell, and this dogma is not at the present continues being a right one. This transition from a differentiated cell to an embryonic-like cell was due to the presence or to the addition to the expression of 4 factors or proteins that are known like the Yamanaka factors. These Yamanaka factors are not only needed for reverse the development or some features of the development, but also we know that could also reverse some features associated to aging. Indeed, in the group of Isidro Ferrer, they describe that there is a decrease in some age-associated hallmarks by partial reprogramming using these Yamanaka factors. In that way, it was found that in old mice, you can find an improved regeneration in organs like spleen, skin, pancreas, or muscle. But nothing was not or was indicated at the present about the possible reprogramming in the central nervous system. In the central nervous system, we know that we can study if there is this reprogramming, but it's very, very large to do very general studies. And then it's better to concentrate in a specific region or area, mainly in a brain region. We choose to study aging in brain, one region, following the so-called the Ribot's law. This law, this is a very old law, indicated that brain structures with a late development may be the first to become old and to degenerate. One of these brain structures is the dental gyrus. And the dental gyrus is important also because not only because its maturation is several days after the birth of, for example, of the mouse, but also it's important because in that place take place adult neurogenesis. And adult neurogenesis is related with episodic memory. It is also known that adult neurogenesis decrease with age. And also is known that it's impaired in humans in some neurodegenerative disorders like Alzheimer's disease. Although about this point will be the next talk by Miguel Flor Garcia, working in the group of Maria Llorens Martin, that he will focus his presentation only in this point. Then our objective in this talk and in our work is to express the 4 Yamanaka factors and to look for possible changes in the age brain to see if we can have some or any reversion. And to do that, we have used as a model the transgenic mouse model obtained by Dr. Manuel Serrano, that was the same model, the same mouse that was used by Dr. Isidro Belmonte to look at changes at the peripheral tissues. By using this model, in which a transcriptional activator, RTTA, that is located inside the ROSA26 locus, and that once that this transcriptional activator is expressed and upon binding to doxycycline could bind to another promoter, the theta promoter, that could activate then a polycistronic cassette encoding the 4 Yamanaka factors indicated in the slide. Then, depending on the administration of doxycycline, we have or not expression of the 4 Yamanaka factors. To look as a markers, to aging markers, to know that we are using real old cells or aging cells as aging markers, we have used some, we have analyzed some epigenetic changes, mainly present in histones, like some methylation in histone 3 and some phosphorylation in histone 3. And when we look what happens with these epigenetic changes during aging, we found a decrease in the histone methylation. We compare, for example, 3 months old with 10 months old dentagyrus mice, and we found a decrease with age of this marker. Also, we found a decrease in histone phosphorylation, but also, and that could be important, we found a decrease in some adult neurogenesis markers like double cortin. Then this is the thing that all the markers decrease with aging, and our purpose is to see if we add the Yamanaka factors, we can reverse the levels of these aging markers. And we did by administration of doxycycline and then inducing the expression of the Yamanaka factors. And our results indicated that almost not any difference in the different markers for adult neurogenesis, starting from the stem cells, proliferating cells, neuronal differentiation, and so on. We didn't find any change in the absence of the presence of Yamanaka factors. However, out of these newborn neurons, in mature dentagyrus neurons, we found that markers like histone methylation were reprogrammed, were changes in the presence of Yamanaka factors, and then the level of methylation in the presence of Yamanaka factor in a 10-month-old mice is similar to that level present in 3 or 6-month-old mice. However, a continuous administration of doxycycline, a continuous expression of Yamanaka factor, results in a decrease in the survival of the mice and the appearance of tumors. There is very toxic, the continued administration of the Yamanaka factor, the continued expression, let's say, of the Yamanaka factors. Then we changed the protocol, and instead of doing a continuous administration of these factors, we did a cycle administration of these Yamanaka factors. And in those conditions, no progress with survival. We don't have, was no progress at that level. And was not also progress with the appearance of tumors. The mice look very healthy in general. And also we found that also was a good expression of the different Yamanaka factor in these conditions, with this protocol. Then we start to see it was or were any differences upon the expression of the Yamanaka factor in these conditions, looking first at the RNAs expressing some adult neurogenesis markers. And no difference were found at the RNA level, and no difference were found on the protein level. Were exactly the same in the presence of, in the absence of Yamanaka factors. However, again, was a clear difference in mature neurons that are in the dental gyrus but are not newborn neurons. Against an increase in the methylation of histones were found. Then the conclusion at this point is not main age-dependent changes related with adult neurogenesis markers can be found. Then, in the presence of the Yamanaka factor, then the question is, what happens with the mature neuron reprogramming? Indeed, we already found that there was changes in the presence of Yamanaka factors in the level of histone methylation, but there are another changes. Then we knew that can be a possible correlation between the level of calretinin plus cells and memory impairment that was indicated some time ago. And then we look at it was we can find any change in the level of calretinin plus neurons outside, but close to the dental gyrus. And we found that yes, it was an increase in these calretinin plus cells. Also, we know that there are NMDA receptor subunit, the GLU-N2B receptor subunit, that is mainly present in young animals, but decrease its level with aging. And then we found that in the presence of Yamanaka factor, there is a reversion of this decrease, and in all mice, there is an increase in the level of this subunit. However, there are not an increase using like a marker, another postsynaptic protein, like the GLU-R1 subunit, that in this case was not any difference in the presence of in the presence of Yamanaka factors. Then we have several changes in mature cells that are due to the presence or to the expression of Yamanaka factors. And we look at if those structural changes could be related to some functional changes. And good functional changes to look for is problems in memory. And doing the object recognition test, we found that in those mice expressing Yamanaka factors, memory is retained after 5 days in those mice that are expressing Yamanaka factors. This memory retention indicated that something is happening in those changes that we found with the Yamanaka factor expression. In conclusion, it seems that under Yamanaka factor expression, there is a mature neuron reprogramming and a memory improvement. Then I would like to finish here. But before finishing, I would like to thank to the people of the group. This is the group 2 years before the pandemic. I mean, some long time ago. It's another time in our lives. And also in that photograph is not the first author of the work, Alberto Ruiz Matellán. And also I would like to thank Manuel Serrano because we did this work that I just told you in collaboration with his group. This work will be published at the beginning of November. Okay, thank you very much. Thank you.
Thank you very much, Jesus. I would like to then get into the discussion forum. Before going into the questions, I just wanted to to ask the three speakers to be online, but only use the microphone if a question is posed to you. Otherwise, we will have some coupling and some interferences and so on. I remember everyone that you can do questions through the chat in the platform, and I will transfer those questions to the speaker. And every speaker also, if you want to intervene in the discussion, just please feel free to do it. Just wait for the other person to finish talking, and then put your microphone on and just intervene. So so far I only received one question actually. So I'm gonna do that and ask everyone to keep sending questions if they're interested in. So the question I received is addressed to Eva Carro, and is posted by Lara Ordóñez, and the question is as follows. Are lactoferrin levels modified in transgenic AD mouse models? Eva? And the question is as follows. Are lactoferrin levels modified in transgenic AD mouse models?
Yeah, thank you so much, Lara, for your question. Yes, indeed, we are performing these studies in the transgenic mouse model. Yeah. And yes, we are finding some differences. We are finding a reduction in the lactoferrin levels also in these transgenic mouse models, but due to the difficult access to this kind of sampling in mouse, we only obtain 20 microliters, more or less. Then we are finding some variability, but in general, we also find a decrease similar to those observed in human patients.
Thanks, Eva. While we wait for some more questions to get in, I would just like to ask the other speakers if you want to question each other or if you want to add something else to the talk, please feel free to do it. Since I'm not getting any extra questions, so if you want to intervene, please. While we wait for some more questions to get in, I'd just like to ask the speakers if you want to question each other or if you want to add something else to the talk, please feel free to do it. I just got a new question, just got in. It's a question to Jesús Ávila, and it's posted by Inmaculada Cuchillo, and the question is as follows. Have you seen any effect of YF in the hippocampus?
Yes, we are now doing this kind of studies. In fact, the antigyrus is present at the hippocampus, it's part in the area, is in the hippocampal area. But our next studies are focusing in that hippocampal area, but also we start to look what happens in different cortex areas And at the present, our findings are that there are different agents in different type of neurons. We like to indicate exactly which are the characteristics of this differential agent, but we are now in the way. Yeah, and then thank you, Immaculada, for the question, but I hope to have new results very soon.
Test other transcription factors in combination with the Yamanaka factors?
Please, could you repeat the question? I was unable to listen to you.
Whether did you test other transcription factors in combination with the Yamanaka factors?
Please, could you repeat the question? I was unable to listen to you.
Now, Jesús. Okay, the question is whether did you test other transcription factors in combination with the Yamanaka factors?
We only tested
other transcription factors in combination with the Yamanaka ones.
Okay, the question is whether did you test other transcription factors in combination with the Yamanaka factors? Not at the present time, although we are trying in collaboration with Spisua Belmonte to change the Yamanaka factor for synthetic small compounds that could replace them. But this is now in the process. This is now a working process.
I have one other question again for Jesús Ávila. This question is posed by Isabel Fariñas, and the question is, how is the expression of endogenous reprogramming factors in neurons of the reprogrammed mice?
This question is posed by Isabel Fariñas, and the question is, how is the expression of endogenous reprogramming factors in neurons of the reprogrammed mice? How is the expression of endogenous reprogramming factors in neurons of the reprogrammed mice? Please, you can give me an example of endogenous reprogramming factors.
Answer to that and send me a message so I can transfer that message to Jesús. In the meantime, we're going to go for another question. This one is posted by Carlos Matute and is addressed to Isidro Ferrer. The question is whether did you check glucose transporter levels in oligodendrocytes in GGT? Another question, this one is posted by Carlos Matute and is addressed to Isidro Ferrer. The question is whether did you check glucose. The question?
Well, we tested not only in oligodendroglial cells.
The question was addressed to Isidro Ferrer and posted by Carlos Matute.
The presence of this marker in oligodendrocytes. So we have studied this marker, and what we have seen is a decrease in the expression of this marker in the neuropile and an increase in the capillaries. So this is an important point because it seems that we have an increase in the glucose transport factor in the endothelia, or at least in the capillaries of cases with GT. So there is an increase in the capillaries and a decrease in the neuropil.
Isidro. So I just got a message from Isabel completing the question she made earlier. So just to remind you all, the original question was addressed to Jesús by Isabel, and it was, how is the expression of endogenous reprogramming factors in neurons of the reprogrammed mice? And Jesús was asking whether endogenous reprogramming factors were referred to, and Isabel now has sent a message saying, I should have said pluripotency factors like OCT4.
Thank you, Isabel, for the question. We didn't test this point yet.
Thank you, Jesús. I don't have any additional question yet, but I would like just to give the opportunity to the speakers if they want to make any additional comment or ask each other at this point. I'm not getting any additional questions. I think it's time to close the session here. So I would like to thank all the speakers for their presentation, for sticking to the time. I also have to apologize for the audio problems we have at the beginning. Hopefully they will be solved. They were solved. And with this, I will close the session, and I will resume the next session at 11:30. Thank you all.